our science project experiment for biological macromolecules, but instead it's divided into two parts. So the first one would be for sugar and starches. Basically, for the first part, which would be for the sugar and starches, the purpose of that would be to find any presence of reducing sugar and starches.
This is all of our supplies. We already started setting up for our first experiment, so we're going to start that now.
Okay. We'll keep you guys posted on the other parts. Alright, so.
Okay.
So right here we have the unknown solution.
And then we already poured our distilled water, 10 milliliters, in this graduated cylinder. So now what we're going to do is we're going to pour it into the unknown solution.
I don't know if you can see it, but it's practically this one.
So we're going to pour it in. And be very careful because we don't want to blow up first. Okay. Now once she's poured, everything. And so when she's poured everything, we're going to put the top on it, screw it back on, and we're going to lightly shake it until it is dissolved. Okay, now for our second part of the experiment, we have an egg. We're going to separate the yolk from the whites and only keep the whites. We're going to put the whites in our 9-ounce plastic cup. That's what we're gonna do now.
Off we go.
Just be very careful to not.
And make sure we don't have any eggshells either.
I think that's fine. So this is our egg whites.
And here you have the yolk that we're not going to use. Do you think 0.5 would be like here? So that's 5, 4, 3, 2, 1.
No, it should be 10, 20, 30. So now that we have our egg whites separated from the yolk, we labeled a test tube EW, and we're going to put 0.5 milliliters of egg white in there.
And then in the meantime, I'm going to go ahead and put 4.5 of distilled water into this graduated cylinder.
So I just put 0.5 milliliters of egg white in our test tube, which is labeled EW. And right now she's measuring out 4.5 milliliters of distilled water,
which is already in the graduated cylinder,
which we're going to pour into our test tube, mix it with the egg whites.
Is there more? No, it's it. Okay.
And then we're going to mix it up. I don't know if there's another way we should be mixing it.
I don't think so. Because they don't have a little set over.
Okay. And this is our egg white tube. Sample tube. So now we have the rest of our test tubes. Yeah. Milk. Unknown. pH2O. And glucose. And we're gonna fill them up with their designated liquids. Yeah.
So what I'm going to be doing in the meantime is measuring 1 milliliter of water into the DH2O2. And as well, I'm going to be adding in 1 milliliter of milk into our milk tube.
And while she does that, I'm going to be putting 20 drops of the unknown solution into a test tube and doing the same with the glucose.
Oh, there's one in here. So we have our milk and our test tube. Now we go to the next step.
Yeah. So now my partner is gonna put 40 drops of the Benedict reagent into the glucose test tube, the unknown test tube, the DH2O test tube, and the milk test tube.
So off we go to dropping 40 drops. So now we're gonna be lighting up the burner fuel to start boiling the hot water. And then, yeah, take both at the same time.
This is taking so I'll just do it halfway. Experiment and we just recorded our data. So it seems like the glucose has the most sugar present because it turned a reddish brown color. We didn't get to record it because we were too distracted, but we recorded the data. So the milk turned a greenish color, so it would be in second place for sugar present. Then the egg whites stayed clear. They're actually still here, so I could show you this one. This is the egg whites.
There was no change. Only thing that the egg whites were getting cooked.
Yeah. And then our water and unknown substance stayed blue. So there was no sugar present in those. So that would be our final conclusion from that experiment. Now we're going to start our second experiment on starches.
We'll see you guys later.
So for our second experiment, we're gonna do 1 milliliter of glucose, starch, unknown substance, water, and then we're gonna add this IKI IKI indicator into each one of them, 20 drops. So we're going to get that done right now in our test tubes.
So as you can see right now, I'm going to go ahead and put the unknown solution into the unknown tube. Okay guys, so far we have already added all the solutions that had to be in the test tube, which was for the unknown, the glucose, the milk, the starch, and the H2O, which afterwards we added 1 drop of, what was it? The IKI indicator, which it's all in there. Right now they're being mixed. We have already cut as well our 1 centimeter piece of potato as well. And now we're gonna put it on a plastic spoon.
And after we put it on that spoon, we're gonna observe which of the colors. This is already changing. The starch is turning a dark color, and see how each one changes. So black is positive for starch, and that like amber, yellowish, goldish color is negative for starch.
We excuse for the background noise. This one's like a paleo right now is gonna drop.
What if it changes?
We're going to drop 1 drop of IKI indicator on the raw potato. Yeah.
Oh, it's turning black almost immediately.
As you can see, the potato turned black immediately with the IKI indicator droplet. Because clearly the potato also has starch. So that's another reason.
Interesting.
So here's how it looks afterwards. Looks like a rotten piece of potato. This experiment for reducing sugars, we concluded that the presence of reducing sugars varies in the colors of the test tubes. So the tubes that were blue were the ones that didn't have a reaction and didn't have any presence of reducing sugars when the water was boiled, as to opposed to the red tube that did have the reaction of reducing sugars, and it was highly concentrated since it was in a dark red color. And in addition to that, we also tested the starch with the IKI indicator, and we noticed that with the potato, it had turned black, meaning that there was a high concentration of starch in the potato, versus the other elements that were in the tubes didn't really have a reaction to the IKI indicator since there was no presence of starch. So therefore, they stayed into the color of amber, or like a light yellowish color. So in conclusion, our hypothesis was correct for the starch, the IKI indicator, which the potato did turn black. But unfortunately, our hypothesis for the testing the presence for reducing sugars, it was incorrect, because we inferred that most likely we would be able to see the difference of the presence of sugars when it's in the tube, maybe thinking like it was probably going to be crystallized while the other substance floats, but instead it just changed color. So we learned a quite interesting knowledge in our experiment. I hope you guys enjoyed our video with biology macromolecules. I for sure enjoyed the experiment, and I would most likely do it again if I had to. Although I do hope to try next time maybe, an experiment that deals with DNA, to just be able to like analyze the little features and little structures of it versus to like seeing it in a textbook.
But overall, so much for watching, and I hope you guys enjoyed our video, and hopefully our video in the future can help little kids or even teenagers or early on adolescents to learn more about science so it can make an impact in the world. So thank you, bye.